Hello, welcome to our group presentation. We are from Group21 and our title is Genome Resequencing and Reannotation of E.coli ER2566 strain and transcriptome sequencing under overexpression conditions.
Hi, my name is AMIRA AQILA. I will explain the background and also the objective in this article. The E.coli expression system is one of the most well-characterized classical expression system for recombinant protein expression. This is because E.coli offers a clear genetic background, fast breeding, low cost, high cell density culture, and high protein expression too. The E.coli ER2566 strain is a common laboratory tool that takes advantage of the expression and growth properties in the B-line strain. But even with the rapid development of biological laboratory techniques and sequencing technologies, there still tend to be incomplete and fragmented data. For example, the annotation of the ER2566 strain was incomplete, with missing gene names, miscellaneous RNA, and uncorrected annotation of some pseudogenes. This is because most genomes are annotated automatically and not subjected to any manual review, also because of limitation in the annotation of the strain. The objective in this article have employed a series of automated annotation and combined this with manual inspections with high throughput analysis to re-annotate the ER2566 genome and other vector genome with high speed and accuracy. The re-annotation and sequencing result in this article will provide a better understanding of some of the biological process of the ER2566 strain and may offer insight into future biotechnological application in bacterial engineering.
Hi, my name is MAIZATUL and I will explain about the methodology. There are 6 methodologies that used in this case study, which is the first one is bacterial strain, plasmids, and culture conditions. From this article, it explains how they get the E. coli B strain ER2566 and how they maintain the quality of the strain before it is used. For the second one is DNA extractions and sequencing. In this study, genomic DNA was extracted by acetyl-methyl-ammonium bromide known as the CTAB-based protocol, and sequencing was performed by the Institute Genomic Beijing. For the third one is RNA extractions and sequencing. RNA was extracted using on the MasterPure RNA purification kit, and for the sequencing was performed on Illumina HiSeq 2500 platform. Next is the genome reannotations of ER2566 strain. The whole genome was downloaded from NCBI reference sequence database. The fifth one is the variance calling whole genome sequencing by read mappings. The quality of the raw reads were determined by using the FastQC and filter it using FastP to remove the low quality bases. Lastly, variance calling whole genome sequence by genome assembly. Draft cortex were created by using the SPADE. The assembled draft cortex and sequence library were used as an input into the redundants.
Okay, next, I will proceed to the result and discussion. So for this study, they do the reannotation of ER2566 genome is to provide a further prediction of the information detail about the coding sequence or non-coding sequence, pseudogenes, and other repeated sequence, as many others. So they do the prediction and identification of coding genes by the prodigal, which is predict 4,160 of a coding sequence, and then they do the reannotation of the coding sequence by the sequence alignment to the sweet prot database and find out that 96.2% is a coding sequence, 3.3% is hypothetical gene, and 0.5 is a pseudogene. And after that, to improve the prediction, they do the reprediction by the gene finder, which is Climber, GeneMark, and ZCurve. And from this gene finder, they found that 428 coding sequence were found. By the Climber is 194 coding sequence, GeneMark 123 coding sequence, ZCurve is 201 coding sequence. And after that, they do the BLAST-P to filter out the coding sequence, and resulting in the 402 coding sequence, which is resulting for additional of 43 genes. So as a result, as in total, 4,066 protein coding, along with a 136 coding sequence for the hypothetical genes. And after that, they predict genes coding for tRNA, RNA, and ncRNA, argon, bernard, and infinite, and infinite and cRNA finder independently is to predict the gene coding for tRNA, rRNA, and ncRNA. And they found that 230 new miscellaneous non-coding RNA and 2 new tRNA. And overall tally of gene encoding for is 87 tRNA, 22 RNA, and 245 anti-RNA. So they also do the comparison of the annotation with the RefSeq annotation due to the trimming and splitting gene in the protein alignment. And totally of 190 protein coding were removed as they were identified as either hypothetical proteins or pseudogenes. 237 new coding sequences were added in the reannotation list. And then this is show the differences between the RefSeq annotation and the reannotation. So in the reannotation, one pseudogene was identified as two genes, which is ins A and ins B, which shows that it shows that strong homology of the insertion element protein IS1. And after that, they are re-identified the two genes, which is like Z1 and like Z2. Whereas the hypothetical protein was reannotated and shown to be highly homologous with the DNA-directed RNA polymerase gene is CBD9006 from E. coli strain BL21 DE3. So they do compare that genome, which is BL21 DE3 genome, with the ER2566 genome, and they found that the genome element of both demonstrate a high degree of consistency. And this figure show the comparison between the two genomes. And the blast result show is proportional to the percent identity of the hit, and 92.3% of CDS were identified in ER2566 annotation as the identical gene symbol. And this reannotation effectively eliminates the possibility of false interpretation introduced by the original annotations, and it provides a more integral view of the regulatory networks in ER2556 strain. And then this is overview of final annotation, reannotation, and BL21DE3 annotation. So miscellaneous RNA shows the increased number from 9 to 225. It's helped to provide a more informative profile about the genome and for the molecular mechanism analysis of the analysis. And next, we proceed to the other results and discussion.
Hi, my name is Yao-Sing Yi. So I will explain about the variant calling of a whole genome resequencing under consecutive subculture. This is to identify any nucleotide level differences. First, the raw read will map in the C2566 reference genome with a good coverage dip. There is no single nucleotide polymorphism or indel was detected. But in the various de novo assembly software, which is used to construct a confined and long scaffold, one inversion was found. This will produce a genome assembly issue caused by a high repetition agent, and this will be corrected by the subsequent central sequencing. Next is mutation detection by the RNA sequencing under overexpression. The function of the RNA sequence is to identify any variation in the ER2566 genome due to overexpression mutation. There are two types of replicants, which is the B37 act as a control sample and Y37 act as an overexpressed sample. A result came out, there are 98% BP rates were mapped in the B37 and 39% BP rates is mapped in the Y37, which is lower than the B37, and this is due to the AMIRA transcription in the engineered plasmid is not required in the genome. The variant detection analysis also be done to detect the mutation. Mutation of the C to T substitution occur in the Y37 sample, nearly about 100% mutation rate. And the mutation also found in the B37 sample as well, with a mutation rate up to 85%. But this mutation cannot be detected by the central sequencing in the bacterial genome. Therefore, we assume that the discrepancy between the transcribed RNA and the genome sequencing may appear from a modification during RNA transcription. Because of the mutation position is being located in the high-efficiency RNA methylation, so the overexpression of the lag I could lead to the increase of the RNA methylation rate.
Now my turn, ZAWINA, to present conclusion and comments. Conclusion. The general scientific community and the biotechnology industry are used the ER2566 strain. The annotation of the E.coli ER2566 strains also uncover a key site that might be involved in the transcription and translation of gene encoding the lactose operant repressor, besides improve the ref sequence data. Our pipeline that we propose might offer a universal method for the reannotation of other bacterial genomes with high speed and accuracy. The study of E.coli ER2566 strain gives us a better understanding of gene function for the ER2566 strain under external burden. Besides, that more clues are provided about engineered bacteria for biotechnological applications. Comments. First, there are 6 methods to study, so we need to understand about each of these methods to make us know how each method are run. Second, the annotation using min CED 0.2.0 software are not being discussed in the article, as how the final annotation of cluster regularly interspace short palindromic repeats CRISPR being done.
And that's all for our group. Thank you for watching. Hope you enjoy it.